I will tell you a little bit about the idea of precision medicine on the one hand, and what possibilities there are when it comes to a partnership, an industrial private and public partnership as the one that we have had in a project called PRECISEADS, which is the one I show you here. We've been recently, it's 10 years since these Innovative Medicines Initiative projects from the European Union started. And for those who are scientists in the public domain, or even in the private domain as biotechnology companies, I would like to tell you that I feel that this is a good opportunity for scientists to put together collaborations. What I see here in this forum is that there are a lot of biotechnology companies with great ideas, and trying to find also contacts in the public and the academic community. I think it's quite good when it comes to trying to apply for funding of this type, which can actually help to move forward some of your ideas. So what PRECISEADS has had as a goal is actually to try to redefine the taxonomy of a number of diseases that are called systemic autoimmune diseases. So what PRECISEADS actually found was that there was an important patient need. Systemic autoimmune diseases are not extremely frequent. They're not like cancer, but they're diseases that affect very young people in general. One of the diseases I've been always very passionate about is systemic lupus erythematosus, which is an autoimmune disease that occurs mainly in women, and actually very young women, whose cost per person is much higher than for any cancer patient. These patients have to go and monitor themselves every 3 to 6 months to the doctor, because in the moment that the disease becomes active, it can actually be fatal. So these diseases in general are estimated to be around 1 to 2% of the population, the systemic autoimmune diseases. Autoimmune diseases in general, if we consider multiple sclerosis, type 1 diabetes, and other diseases, reach about 5% of the general population altogether. They are lifelong debilitating diseases, chronic, and as I told you before, many of them start very early, in the 20s or the 30s. So you can imagine a person that has one of these diseases is going to be all her life, mainly women, also men, suffering from this disease that have no cure, but they have only symptomatic and very general type of treatments. For instance, lupus and another disease is called scleroderma or systemic sclerosis, which is the fibrosis of the skin or of other soft muscles, it can also be, many prognoses are much worse than for many cancers. So 1 in 5 people with lupus actually develop the disease as a child, mainly during the teenagers. And I can tell you that, for instance, and this is data that I just recently got in the United States, around for Hispano-Americans, people of Latin American origin that live in the United States, it's the 5th cause of death in women, lupus in this case, and the 11th cause in European American women. So I don't think there are diseases that we should actually look on the side. I'm telling you this because their funding is really very scarce, and this is only one of the few projects that there are that actually support this type of diseases. Many of these diseases, as systemic autoimmune diseases, have the main problem is that they cannot be easily diagnosed. And many of the patients actually spend years without a clear diagnosis. In fact, some of these individuals are actually called undifferentiated systemic autoimmune disease or undifferentiated connective tissue disease because they're never clinically diagnosed because they do not fulfill a number of criteria that for a number of years physicians have been trying to put together. So each patient diagnosed with each condition is basically different. They are terribly heterogeneous. Their treatment today is really by trial and error. And getting the right treatment to the right patient that is precision medicine would be actually something that would transform the possibilities for these patients. And this is basically the aim that we have had. So the diseases that we have been working with are rheumatoid arthritis, lupus, Sjögren's syndrome, which is a disease mainly infiltration of lymphocytes in the salivary glands and lacrimary glands that affect a lot of women of middle age, more or less. Scleroderma, as I told you before. Lupus is autoantibody production where you have kidney disease, but also skin disease and many other manifestations that are very heterogeneous, and rheumatoid arthritis that can cause actually erosion and complete deformation of the hands and the feet and the knees and other joints. So our main idea has been actually to try to see whether we can create a new taxonomy of these diseases. So we definitely started with a very big challenge where we recognized that patients differ from one another, that they suffer from different diseases, that have different prognosis, that react differently to drugs, but that the clinic cannot tell how each patient will actually progress. So there was a need of precision medicine that we actually, there was actually a call on precision medicine with this article that was published in Nature Drug Development, and that we tried to respond into in this particular project. The project, PRECISEADS, is a collaborative European project that has actually public and private partnership. These are all the public partners, as you see, from all over Europe. These are the main private partners, Bayer, Sanofi, Servier, UCB, Lilly, and then a couple of, one Spanish and one Swiss, small biotechnological companies. And so it's a joint initiative that actually has a funding that has, these type of initiatives are partly funded by the European Union, half of the funding, and the other half comes from the pharmaceutical companies, either in kind, which is the most typical, or sometimes also in cash. Or they directly can actually pay for a number of assays and do a number of things in-house. One thing that I have learned about this project in particular is confidence and trust, which is something that we academicians actually lack in general. I can tell you that. We academicians, you probably know how we work. We're always afraid someone's going to copy us, is going to publish before us. But here we have actually put together a project where we all trust each other and we're working really as a team and moving forward as a team. This is one of the meetings that we had actually at our center in Granada. So building a taxonomy was simple in principle, really, because what we thought is that, well, the current taxonomy divides diseases into these diagnoses. That is how the doctors diagnose these diseases according to clinical criteria that have certain limits. And as I tell sometimes, is that the doctors go on trying to reclassify the diseases using their clinical data, but they don't realize that they will probably cannot look beyond what is maybe in the serum or in the skin. They cannot look inside the cell, or they cannot look inside the DNA, of course. And so the new taxonomy of individuals would be based on a molecular signature where we simply say that we put them all together and then try to cluster them again. That's the basic principle of this project. So this is, once again, we get these individuals with the different diagnoses and the diseases. We put the study design is to put them together and then to do a number of molecular parameters that we have been studying, and then we recluster them and trying to find this new classification. And of course, this sounds very simple, right? But it's not so simple. So our aim, and I have to say that because of the different layers of molecular analysis that we were studying, we really couldn't tell how many individuals we actually needed. So we decided to be ambitious. Why? Because you cannot statistically define how many individuals you're going to have. You're putting together transcriptome data, you're putting together epigenome data, you're putting together metabolome data, and because there are different layers with different number of features, it's very difficult for you to actually say, oh, statistically this is how many I need in order to make the clusters. You can't. Okay, this is very different from what we used to do before, for instance, in genetics, for example, right? So we actually had two cohorts, a cross-sectional one, a cross-sectional set of patients, and an inception cohort. This has just been recently, we finished the recruitment and the third time point recruitment, because this is a follow-up. And here actually we had two phases, one early phase where we did a more in-depth evaluation in order to use many of the things that we found useful here into this larger number of patients. So in the end, we basically have around 2,000 patients in the cross-sectional cohort and 666 controls. And 200 patients that have been followed in an inception cohort, which means patients that have less than 1 year since diagnosis and then followed at different time points and see to what extent, if we can actually validate the clusters that we find in this cross-sectional cohort, and what changes treatment actually has on this on the clusters that we find. Although, of course, we have treatment as well, and this is something that we take into consideration. So it wasn't easy. We elaborated a number of protocols for multi-center flow cytometry analysis, patient recruitment, centralized sampling and processing, sending to the labs, upload the analysis, everything centralized. And actually everything coming out from here and going out and coming in. So what we did was really, for instance, the different centers are all across the place. We needed actually centers to have flow cytometers in order to take fresh cells. For instance, Berlin received samples from Hungary and from Vienna. We received samples from Malaga. In Barcelona, they received samples from Santander. And so the different groups, when it came to the flow cytometry, when fresh cells were collected, it was like this. Then all those samples that were collected and were sent back to Granada, to the Andalusian Biobank node in Granada. And then the Biobank actually sent to the different laboratories. Some things were done in Granada, other things in Barcelona, other things in Stockholm, some other things, where else was it? Not in Berlin, but different things, well, in Brest, and so on. All that data was put together into a single server, which was centralized in Switzerland, where the company Quartz Bio actually took care of all the data, still taking care of all the data was uploaded. And then this data, and according to all the privacy laws and privacy rules that are necessary, is being downloaded by those groups that are actually performing the analysis. And I have to say that the primary analysis are ongoing here in Granada. Okay, so achievements. So we recruited basically 99.3% of all the patients that we needed. Actually, this was just a few months ago when we presented this at the IMI presentation. We have done multi-omic analysis, which have never been performed before in autoimmunity, in autoimmune diseases. We have had over 100,000 samples from about 3,000 donations that have been registered by the Biobank in Granada. And these are some of the analyses that have been done together, sequencing of the RNA, genotyping and HLA imputation, actually not genotyping, the unique standardization of the flow cytometry analysis across 11 different instruments that we were able to do. And this is also quite unique. We were able to harmonize the flow cytometers of different sorts throughout 11 different types of centers and 11 different types of instruments. So this is more or less the distribution of the patients and the samples that we have. But then to the results, and this is still ongoing work, actually what I can present to you. And what I can show you is that here you have this plot over here, which shows you each of the colors here is one of the diseases. You have here lupus, scleroderma, rheumatoid arthritis, Sjögren's, primary antiphospholipid, mixed connective tissue disease, and the undifferentiated connective tissue disease. And what is very interesting, we have now done this in 2 independent sets of patients, and we have validated it actually in a statistical, very correct way and very stringent way. And what we show here is that you see the diseases following into these different molecular clusters. And this has been done. We see we have done adjustments for the treatment. We see no cluster that is due to, for instance, treatment, or no cluster that is due to, for instance, the place where the samples were actually from, or batch effects of any type. So what we show here is that you have some groups where you have some diseases that dominate a little bit more, but that they are found within this. So what is this useful for? Well, basically it tells us now that their patients actually can fall into different types of groups, that are molecular groups that are different. And by knowing this, the next step will be actually to see if we follow the patients longitudinally, to what extent does the treatment and the time of their relapses and remissions that they have in their diseases may actually change or not this particular picture. And then to see whether any of these patients eventually would be better or non-responders or no-responders, for instance, to a given treatment that they're given at a certain point. This would give us also an idea already. I can tell you that one of these clusters has to do with the very important gene expression signature that patients with lupus and Sjögren's actually have. So there are a few patients that fall within that signature, but not all. While the other patients, for instance, another interesting group is one formed by two completely different diseases from the clinical point of view, like rheumatoid arthritis or scleroderma. And what we see is that these individuals fall into a similar group. How come individuals from two very clinically different diseases actually fall into the same clinical group? So as I told you just before, we have validated this independently in another set of patients. These groups, we have it in another set of patients. And so what we're doing now is actually writing a publication. So of course, that is something that we scientists are very interested in doing, of course. And so, so in summary, we have managed to actually done something that it's unprecedented in the field of autoimmunity, which is really conducted a number of multi-omic analysis. What I showed you here is primarily the transcriptome and the methylome data, but we have now been also integrated. Now that we have these clusters, what we're doing is characterizing these clusters for a number of markers, from serum markers, serological markers, metabolomic markers, and other sort of markers to see which are the best markers to distinguish these different groups of patients, okay? So this is actually enabling precision medicine at the first stage. Currently defining biomarker signatures within the patients with these diseases that will empower, we believe, physicians to tailor therapy for the individual patients. Just to give you a more concrete example is, for instance, one of the treatments that is coming up today in lupus, for example, which is an anti-type 1 interferon receptor treatment. A phase 2 trial actually worked very well when they divided patients into interferon high and interferon low. But not all patients with interferon fall into these groups, okay? What they did in the first and the third phase trial was that they put them all together and the trial failed, okay? So here is actually really like, come on, why didn't you separate them again, or try to do something to see really which are the patients that will respond and patients that will not respond. And it seems that by grouping the patients, obviously you're diluting so much an effect that you're not going to be able to see what is really good for those patients. Maybe not all of them are going to be able to receive it, maybe a few. And if we start actually putting this type of work together and doing and determining this type of clusters, we might be able actually from the start to say this is a patient that will benefit from this type of treatment and this is a patient that will not benefit from this type of treatment, instead of going on making them, you know, test one drug after the other, after the other, after the other without any success. So this project is really just the first step for building towards making precision medicine in autoimmune disease a reality for patients, doctors, and payers within, hopefully, the next 5 years. And we're hoping that we are able to continue this particular work in order to actually be able to answer those questions, like how are responders, not responders, which are the patients that may relapse or remit, which are the patients that would develop a more severe disease, which are the patients that actually are going to best benefit for a certain new biological or new drug that's in the market. So this is our webpage, PRECISEADS, and this is just to show you UCB is the main co-leader of the project together with us in Granada. And this is my group here in Granada, together with this is from the bioinformatics unit and all the people, in particular Guillermo Barturen, who's the one who's been doing the clustering analysis together. And this is the last meeting that we had in Ascot in London of the whole project. And I have to thank all of these people because they have been really working towards one single goal by recruiting their patients, putting together the samples, sending their samples, doing the analysis in order for the bioinformaticians to be able to analyze. And we've had fantastic brainstormings that have been really, really very good for us to reach our goal to the maximum. And the funding, as I said, from the Innovative Medicines Initiative, joint undertaking, their grant agreement from the European Union. And if you have any questions, please go ahead and ask. Okay. Come on, I'm really ready to receive any questions. Huh? Don't be afraid, it's actually, I remember when I was young, I actually was very much afraid of asking questions. And I think one doesn't have to be afraid of asking questions, okay? So if you have any doubts, it doesn't matter, you can ask, okay? You have no doubts. In Spanish? No? Sí. Ah, Paul.
When you look at DNA sequence data, of course, you get information. When you look at RNA expression, you also may be able to get information on allele-specific expression, which would be very interested in autoimmune diseases. Now, I was curious, when you look at the DNA methylation data, I presume that you're losing allele-specific information?
No, not really. We are actually using more general information of CpG methylation in general. What we're using really the methylation, the allele-specific methylation is actually for a cross-quality control that we're doing together with the RNA sequencing data. But then, you know, the clustering analysis is quite peculiar, and there are some very interesting results that we're getting. We're looking into the genetics themselves. But not allele specific. I don't see how that would actually give us other sort of information. Maybe yes, but I mean, we're doing QTLs, of course. Quantitative trait loci, for instance.
Directed at the fact that we know there is allele specific transcription at many loci.
Yeah.
But it's difficult to correlate that with methylation.
Yeah, no, but what we are, I mean, one of the things that we want to do is to take the clusters and see how QTLs are actually distributed, if there would be any differences between the different clusters when it comes to the risk alleles and the QT and the quantitative trait locus analysis, for instance, MEQTLs or EQTLs. Okay?
Thanks.
Okay.
Yeah, very nice talk. You mentioned that you were looking into longitudinal studies also. Is that something for the future? Do you have money for that, or is that a new initiative?
Well, we have a small inception cohort that we're looking into, but we do want to perform a larger longitudinal follow-up. We are actually right now working on a proposal, also from the European Union, in order to be able to continue this. So we're hoping that we are going to be able to continue, but I cannot tell you much more. But I can tell it in private, so to say, if you want. Now, if you're interested in investing, that's also welcome, of course. Okay, if there are no more questions, thank you very much for this invitation, okay?
Thank you so much, Marta. We will